Hello, this is Lias Rahal again, and today's lecture will deal with how viruses replicate, so what we basically call the viral life cycle. Um, as usual, you'll see my contact information at the end of this presentation, so make use of that if you have any questions. So why do we want to study the viral life cycle and how viruses replicate? This is quite relevant to us in the medical field because this is how people design drugs. They target the replication of a virus, as you will see later on in this course. The viral life cycle consists of 6 major steps, which we will go over thoroughly. So the first one is referred to as adsorption or attachment. This is when virus attaches to a cell via a receptor on the cell that recognizes a certain protein on the surface of. Remember from our lecture on the elements that compose a virus. If it's a naked virus, doesn't have an envelope, then usually the capsomere is recognized. Whereas if it's an enveloped virus, it has glycoprotein spikes, and these will bind to a receptor on the surface of a cell. And then what happens next is the virus will go inside the cell. This is called penetration or entry, and then the virus is uncoated. And then biosynthesis and replication happens. So replication here in the sense that we're talking about genomic replication. So biosynthesis forming the macromolecules and the proteins that form the virus. And then what will happen is the virus will assemble, which we call assembly or maturation, and then release. Starting with the first step of the viral life cycle, which is adsorption or attachment. So if it's an envelope virus, it should have these glycoproteins or glycoprotein spikes. And then, depending of course on tissue tropism, term that we earlier explained to be kind of like the appetite of a certain virus for a certain tissue or a certain cell type. So if a glycoprotein is recognized by a receptor on the surface of the host cell, then it will bind there, which will later on permit infection. If it's a naked virus, then it attaches to its target cell type or target tissue type via the capsomeres, which compose basically the viral capsid, which is this shell that houses the nucleic acid component of the virus. The virus is now attached. Now it needs to enter. So how does this happen? If you have an envelope virus, what could happen? We basically have 2 mechanisms by which the virus can enter. The first being envelope fusion. So remember, again, from our lecture on the components that form a virus, we said that viruses could have this envelope, which is a lipid bilayer membrane, and it is derived from the host cell membranes or host cell organelle membranes, either the cell membrane or the organelle membranes. So because of its nature, because this is a lipid bilayer membrane, it can fuse with the cell surface membrane, and then what happens next is you release the nucleic capsid inside the cytoplasm. So again, the glycoproteins have attached to the receptor, and then what happens next is fusion of the membrane that forms the envelope with the cell membrane, and the nucleic capsid goes inside the cell next. The other possible mechanism is that after we have this interaction between the glycoproteins on the envelope and the receptors, what could happen next is that the whole thing enters the cell in an endocytic vesicle. So the second possible mechanism by which an envelope virus could enter the cell is endocytosis. If you have a naked virus, again, we have 2 possible mechanisms. The first being what we call capsid reconfiguration. So after the receptor interacts with the capsomeres on the surface of the capsid, what will happen is reconfiguration of this capsid, some alteration in its form that allows the entry of the viral genome by itself into the cytoplasm. The other possible mechanism is that after the capsomeres interact with the receptors, what could happen is you trigger endocytic mechanisms that permit the entry of the entire virus within an endocytic vesicle. Alright, so take a moment to think about this question. I'll walk you through it first. So Richard is a virologist and he's studying the life cycle of a newly discovered virus. He uses a preparation of the virus in which the viral capsid proteins have been radiolabeled with S35. So now that we have the capsid proteins of the virus and they are labeled, they are radiolabeled, so we can trace them. We can trace where they're going. We can trace where the capsid proteins are going. The total CPM is just a measure of radioactivity. It is the counts per minute. So the total CPM of the preparation is 10,000 counts. So we know that we have 10,000 counts of this radiolabel. And then after addition of the virus to an experimental cell line and allowing viral propagation, Richard monitors the counts in various components of his cells. So he's basically trying to figure out where did his radiolabel go. So remember, the capsid proteins are labeled. So the first component is the culture medium. The second component is the outer cell membranes. And then we have the nuclear extract and nuclear membrane. And the last preparation he has is cytoplasmic extract plus organelle membranes. So basically the question wants you to figure out where the capsid proteins ended up based on the CPMs. So where did the radiolabel go? After you examine that, try to figure out where the virus went. So, and what kind of virus it is. So the virus is probably A, an enveloped virus that enters by endocytosis, B, an enveloped virus that enters by envelope fusion with host cell membrane, C, this is a naked virus that injects its genome into the cell after capsid reconfiguration, or D, a naked virus that enters the cell by endocytosis. I'll give you a moment to think about this question and we'll go back to it. Alright, so the capsid proteins are labeled and most of these ended up on the outer cell membranes despite having had a good viral propagation that occurred. So because we have the capsid being left on the outer cell membranes, all you have to do is basically go back and look at the pictures in the previous couple of slides and you'll see that this only occurs in one case, and that's when you have a naked virus, and that naked virus will inject its genome into the cell after capsid reconfiguration. If we had endocytosis, be it whether this was an envelope virus that went in by endocytosis or a naked virus that went in by endocytosis, in both cases the capsid would not be on the outer cell membranes, it will be inside. On the other hand, if you had some kind of endovirus that entered by fusion, then again the capsid will also end up being within the cell itself. The third part of the viral life cycle is movement to target site and release of the genome, which we call uncoating. So this part of the life cycle of viruses is something we don't understand quite very well. We know that there's some interaction with cytoskeletal elements. We know there's some mechanisms that occur. We know some enzymes play a role here, but we don't understand it as well as we understand other aspects of the life cycle of viruses. So let's start with the case of an envelope virus that came in an endocytic vesicle. How is it released? What could happen is the endocytic vesicle will move to the target site after it goes in. How exactly it is guided is not always understood, but we know that the endocytic vesicle will go to wherever the site of the replication of the virus is, or wherever the virus needs to be. And so what will happen is here in this case, this is an envelope virus. Again, this is a lipid bilayer membrane, and the endocytic vesicle is a lipid bilayer. And so what happens is um, we'll have a fusion of the viral envelope with the endocytic vesicle, and then your nucleocapsid is released into the host cytoplasm. The second case is that of a naked nucleocapsid that's in an endocytic vesicle derived from the previous steps that we had. And so what we have here is either the endocytic vesicle will move to its target site, to the viral target site, and then what will happen is lysis of this vesicle, and the nucleocapsid is then released. So what triggers this lysis? It's typically pH changes, sometimes enzymes play a role, and so the virus will basically control these conditions in addition to manipulating some of the cell systems that will permit this to happen. On the other hand, if you had a virus that went in there within an endocytic vesicle, besides the ability to lyse the endocytic vesicle, what could happen is another sort of like capsid reconfiguration that happens within the endocytic vesicle, and then we have release of the genome. So here we're not really lysing the vesicle itself, but some conformational change happens with the capsomeres, and so this leads to formation of a pore, and now the genome is released. Now, if you have a nucleocapsid, this could be derived from the previous steps of entry, such as that of the lysis of an endocytic vesicle that contained the nucleocapsid, or this could be derived from an envelope virus whose envelope fused with the endocytic vesicle membrane, and now this was released. What needs to happen next is degradation of the nucleocapsid and release of the genome. So now we have the genome in the cytoplasm, and this is the site of replication for most RNA viruses. And the reason why most RNA viruses, but not all of them, replicate here is, well, the ribosomes are here in the cytoplasm. And you'll see that most RNA viruses use their own RNA polymerases. You'll see that later on in the lecture. And so they don't need to go to the nucleus. They do not need DNA polymerases. Or our nuclear machinery or stuff that's found in the nucleus. So most of what they need could be right there for them in the cytoplasm. Now, there are exceptions to this. There are some RNA viruses that go to the nucleus and have part of their replication cycle there. So there are exceptions. Just like there are exceptions for some DNA viruses. So most of them go to the nucleus, but there are exceptions. As you'll see, there are always exceptions in science, and that includes virology as well. Now, if you have DNA genome, most of them, not all, most of them have to gain access to the nucleus because they require the machinery there. So in some cases, the viral genome will simply go through the nuclear pore. In other cases, the nucleocapsid will interact with the nuclear pore, and then the viral genome is released inside the nucleus. And still, a third option is that this only happens with very small viruses whose nucleocapsid is quite small, and these maneuver their way through a nuclear pore, and what will happen is that the capsid is degraded there in the nucleus and the genome is released. So there are multiple ways by which the genomic material of a DNA virus gains access to the nucleus. That's because the majority of DNA viruses go to the nucleus and their site of replication is there in the nucleus, whereas most RNA viruses, they replicate in the cytoplasm, but there are always exceptions. Now we'll get to the biosynthesis and replication step. So by replication, I mean genomic replication, biosynthesis making the macromolecules that the virus might require for its life cycle. Now bear in mind, this is probably the most studied step. People know a lot about these steps and what happened there, and most of the drugs that are available on the market target this very step. target the biosynthesis and replication step. This is the step that includes formation of the components of the virus. So if you can stop that, that's probably a good way to stop the viral life cycle. So let's start with the replication of a double-stranded DNA genome. So when we talked about the classification of viruses, we went over the Baltimore classification, which classifies viruses based on their genomic material and how they replicate. And we'll just follow that very same sequence here. So starting with class 1 of the Baltimore classification system with double-stranded DNA viruses. So these viruses, they have a positive strand and a negative sense strand. Remember, again, this is relevant for this lecture as well. When I say positive sense, this means that it has the same sequence, just like an RNA that can be translated by a ribosome, of course, replacing the uracils with thymidine. Now, on the other hand, we have the negative sense strand, which is complementary to an RNA that can be translated by a ribosome. And so this terminology, we will be using it throughout this lecture here today. All right, so in the case of a double-stranded DNA virus, in most cases, they use the host DNA-dependent RNA polymerase to form the RNA component, and then this RNA component can be used to make the viral proteins. And they also, most cases, they use the DNA-dependent DNA polymerase, so the DNA polymerase, to replicate the entire viral genome. Now, there are, of course, exceptions with some big viruses that actually use their own DNA polymerase or RNA polymerase. So one example might be the herpes viruses. So herpes viruses, they have their own DNA polymerase. So they are DNA double-stranded viruses, but they do not use the host DNA polymerase. They use their own DNA polymerase. Another example is pox viridae, or the pox viruses. They also do not rely on host polymerases. They use their own polymerases. And so there are exceptions to everything, but in general, double-stranded DNA genome viruses, they use the host RNA polymerase and the host DNA polymerase to make their proteins and replicate their genome. In the case of a single-stranded DNA genome, so this could be a negative sense or a positive sense. The cartoon here only shows a positive sense single-stranded DNA genome virus, but this case applies to both of them. So what happens here is, again, this is a DNA virus. It prefers to use the host DNA polymerase, and whether it is the positive sense or a negative sense, in both cases, the DNA polymerase can read that. It can form the complementary strand. You get the double-stranded intermediate, and then this can be unwound in order to reform the original genome of the virus. On the other hand, what will happen for formation of the proteins is that because we have this double-stranded intermediate, the negative sense strand can be used by the RNA polymerase to make the RNA, the mRNA, the messenger RNA, that can be then translated in the cytoplasm to form the viral proteins. These viral proteins are then shuttled back inside the nucleus, where we can reform the entire virus itself. So whether you have a positive sense or a negative sense DNA genome, you'll have this double-stranded intermediate that can be used to make the single-stranded genome of the virus itself, in addition to making the mRNA that will encode for the viral proteins. As for RNA viruses, you'll see that most of them, they have their own RNA polymerase. So they do not really use that of the host. That's in most cases. Again, there are always exceptions. So when it comes to double-stranded RNA genome viruses, these have a positive sense and a negative sense strand. So the viral RNA polymerase will use the negative sense one to make the messenger RNA template, and that can be read by the ribosomes, by the host ribosomes, to make the viral protein. and at the same time the viral RNA polymerase can be used to replicate the genome of the virus, and so we have reformed the genome of the virus. Again, something to bear in mind is that we are using the viral RNA polymerase. As for single-stranded positive sense RNA viruses, they have a sequence that resembles that of the mRNA. This can be immediately read by the ribosome in the cytoplasm. And so what we have is translation of the viral proteins, and one of these is an RNA polymerase, a viral RNA polymerase. This RNA polymerase will then allow the formation of the complementary strand, the negative sense template, that can be used by the RNA polymerase to make more of the genome or to make these positive sense RNA strands that can be read by the ribosomes to make more of the viral protein. So this positive sense template can be used for two purposes basically. So keep in mind that single-stranded positive sense RNA viruses do not need really the RNA polymerase to come into the cell with them, and this is why in the case of these viruses it is not typically packaged into the virion or the viral particle itself. This is unlike negative sense RNA viruses and double-stranded RNA viruses that in most cases they come into the cell already having the RNA polymerase, the viral RNA polymerase already packaged, and that's because they want to hit the ground running. So they come into the cell, and the viral RNA polymerase can do perform the various steps required for the viral replication. Whereas the single-stranded positive sense ones, they can rely immediately on the genome to be translated by the ribosomes. That's because their sequence can be read by the ribosomes. So for the single-stranded negative sense RNA viruses, they also require the viral RNA polymerase, but unlike the positive sense ones, they usually come into the cell already bearing that. So the viral RNA polymerase will use the negative sense template, which cannot be read by the ribosome, to make the positive sense complementary strand. And now this positive sense complementary strand can be used to make the viral proteins. It can be translated by the ribosomes, and at the same time the positive sense strand can be used by the RNA polymerase as a template to make the negative sense RNA. Now, of course, there are double-stranded intermediates here, but they're not depicted here in the cartoon. The viral RNA polymerase and some of the enzymes, they can unwind that and reform the negative sense template, or just separate that to make the mRNA, the messenger RNA. As for single-stranded positive sense RNA viruses that have a DNA intermediate, there is a particular family that's quite famous for this. This is the Retroviridae, and this family includes the human immunodeficiency virus. So they have a very special enzyme that's called the reverse transcriptase. So these are positive sense single-strand RNA viruses. Now the reverse transcriptase is an enzyme with multiple functions. The first one being it's a DNA polymerase. So this positive sense RNA genome results in or is used as a template to make a double-stranded intermediate that is not shown here on the picture. So there is a double-stranded intermediate that's composed of a negative sense single-strand DNA and a positive sense single-strand RNA. So the reverse transcriptase has multiple functions, and one of these abilities or functions is being an RNase H. So the reverse transcriptase can degrade the RNA template in a double-stranded intermediate so that we would have this single-stranded DNA. So again, just to recapitulate, the reverse transcriptase, this enzyme has multiple functions or multiple abilities, the first of which being a DNA polymerase. This allows the formation of a double-stranded intermediate that includes one RNA template and one DNA template, so or one DNA strand and one RNA strand. It is also, so this very enzyme, the reverse transcriptase, is also an RNase. It has the ability to degrade the RNA in the double-stranded intermediate. and what is released is this single-stranded DNA template. This very same enzyme has a third function here. It also acts as a DNA-dependent DNA polymerase. So it is a DNA polymerase that can use either an RNA template or a DNA template. So in this rather third step, after degradation of the RNA, what this reverse transcriptase does is form the positive sense DNA strand. So now we have a double-stranded DNA intermediate. Now, this double-stranded DNA intermediate, as you will see later on when you discuss HIV, and when you study HIV or when you study the retroviridae in this course, this double-stranded intermediate can be integrated into our genome. It could be in a quiescent cell, but then we could have reactivation. So in the case of reactivation after a quiescent period, what happens is that the host RNA polymerase will then be used to form the RNA template that can serve either as the messenger RNA to make the viral proteins or the RNA template that actually composes the viral genome itself. Now the last class is that of double-stranded DNA viruses that have an RNA intermediate in their life cycle, and there's only one family that we know that undergoes this. These are the Hepatnaviridae or the hepatnoviruses, which includes only one virus, which is hepatitis B. So in this case, we have a double-stranded DNA genome within this virus, but it is a gapped genome. So the virus goes into the cell, and as you can see here, there are gaps. It's double-stranded, but it's not a perfect double-strand template. There are gaps. There are blank spaces. So this looks like broken DNA to our cells. So once the genome is in the nucleus, our cells do not like broken DNA. We like to repair that. And so what we think happens is we have this repair by the host, by some host polymerase. This is why we have a question mark here. We're not entirely sure of who does that, what enzyme does that, and how this happens, but we believe we have our own repair enzymes that repair the gap DNA to form this beautiful double-stranded circle. Now, next, we have the host polymerases, which will allow RNA transcription. So we have the formation of multiple templates, some of which are messenger RNA, and these have to go back outside to the cytoplasm where the ribosomes will pick it up, will pick these templates up, these messenger RNA templates up, and form the viral proteins. One of these templates is a very long RNA strand that actually includes all of the information of the genome. And one of the proteins is actually a reverse transcriptase. So one of the templates is this RNA strand, this rather long RNA strand that includes the information of the genome. And we also have the reverse transcriptase. And so what will happen is that this RNA is then reformed into the DNA, which is basically the genome of the virus itself. So you've reformed the virus, and then the virus can go out. So people sometimes forget that there are viruses besides the retroviridae that have reverse transcriptase, and one example is the hepatitis B virus or the hepatnoviruses. So this should be a quick and easy question. So I think we've talked about this. Let's see how much of this you've retained at this point. So a viral RNA polymerase is packed into which of the following types of viruses? I'll give you a moment to think about this. Right, and the answer is double-strand RNA viruses and negative sense RNA viruses, and that's because positive sense RNA viruses do not really need the viral RNA polymerase to come into the cell with them, because their genomes will be read by the ribosomes to reform or to form viral proteins, including the viral RNA polymerase that can then be used. Assembly and maturation is the next step. So if you have an envelope virus that is then released by budding, so this means that it will bud out of the cell membrane, what will happen is we form these glycoproteins, the viral glycoproteins, and these are sent to the cell membrane. Now remember that we have what we call the matrix proteins, and so next what will happen is the matrix proteins have an affinity to the capsid proteins in addition to an affinity to the um, envelope glycoproteins. So this will bridge things together. So if you remember the question that we talked about last time about a mutation in the matrix proteins in a disease called SSPE, so in that case we had a defect in the matrix proteins, and those proteins were defective. This means that the capsid cannot really find the glycoprotein for the virus to bud out, which leads to neuronal damage. If it is an envelope virus that will be released by exocytosis, then the glycoproteins are actually sent to the endoplasmic reticulum rather than to the cell membrane. What will happen next is that the capsid will bud into the endoplasmic reticulum, hence acquiring its envelope, in addition, of course, to the glycoproteins embedded within the envelope. And then what happens is that the viral particle, along with its membrane, will go through the Golgi network, and then it will exit from the trans-Golgi, acquiring an exocytic vesicle. So in addition to the membrane that it acquired from the ER, this will become basically the viral envelope. It also acquires an exocytic vesicle that will allow exocytosis in the next step. If we're going to have just a naked virus, there's no envelope there. All that will happen is that the capsomeres and other proteins, what will happen is that things will oligomerize around the genome, reforming the virus. Take a few moments to think about the following question. So we have capsids from the Coxsackie virus A24, and these were added to the genomic material of a reovirus, a picornavirus, an orthomyxovirus, and a paramyxovirus. And each of these was placed in a separate tube. We allowed oligomerization, and so we have the formation of a composite virus. And then these were each used in cell culture with appropriate cell line that can be infected, and this was left for 48 hours. So after this culture period, which of these genomes is most likely to allow infection and to yield progeny? The reovirus, the picornavirus, the orthomyxovirus, or the paramyxovirus? Now at this point, you may not recall the different types of genomes from the electron classification of viruses, but I'm sure that as you go along in the course, you will remember these genomes and types of genomes that viruses have, because this is part of studying each and every virus family. So as you go along in the course, I'm sure you will remember these. But just to help you along for now, so the reovirus, so the Reoviridae, they are a family of double-stranded RNA viruses. The picornavirus is a positive sense single-stranded RNA virus, and the orthomyxovirus is a negative sense RNA virus that has a segmented genome, and the paramyxovirus is a negative sense single-stranded RNA virus. Right. So if you've answered the picornavirus, you've answered correctly, because the positive sense single-strand RNA viruses, their genomes can be immediately picked up by the ribosomes, and this will allow formation of proteins and enzymes required for this. On the other hand, if you just have capsids with the genomes, there are no enzymes included. The reovirus, the orthomyxovirus, and the paramyxovirus, all of these, they are either double-strand RNA or negative sense single-strand RNA, and so they will need their own polymerases, their RNA polymerase, to be packaged into the the viral particle for them to maintain an infection, or to start rather an infection. And so the correct answer is the picornavirus, because it is a positive sense single-stranded RNA virus. Now we get to the release of a virus from an infected cell. If we have an enveloped virus, it can exit the cell by two possible mechanisms. The first one being budding. And so what's going to happen here is that, like I said earlier, the matrix proteins, they will allow the capsid proteins to kind of find their way to the viral glycoproteins that are present on the cell surface or within the cell membrane. And then what we'll have is budding out of the cell through the cell membrane. The other option is exocytosis. Again, the matrix proteins guided the capsid shell to the ER membrane, and then what's going to happen is that, as you probably saw in the previous slides, what we had was budding into the ER, then going through the Golgi network and then exiting. So what has happened towards the final step of that procedure, going through the ER and then the Golgi network, is that the virus acquired this exocytic vesicle from the trans-Golgi. And so what's going to happen next is that this exocytic vesicle is also a lipid bilayer, and so it will fuse with the cell membrane, hence releasing the envelope virus to the outside. And so we're left with naked virus, and how it will exit the cell now is basically by cell lysis. Most naked viruses, they just cause the lysis of the cell. Now, there are some exceptions, there are quite a few exceptions, whereby this viral particle exits the cell by one mean or another without resulting in lysis, but the majority of these viruses, they cause cell lysis. One quite famous example in the field is hepatitis A. So the hepatitis A virus is a naked virus, and it exits the cell without really causing much liver damage, because hepatitis viruses, they infect the liver, and hepatitis A doesn't really cause lysis, but still the virus exits the cell. So not all of them will cause lysis, but the majority will do that. This question requires that you recall what we discussed regarding chemical inactivation of viruses. And so upon addition of ether, Laura's newly discovered virus loses its infectivity. How do you think this virus exits the host cell? The most likely mean is A, cell lysis or exocytosis, B, exocytosis or budding, C, budding or cell lysis, and D, cell lysis. Alright, since Laura's virus seems to lose its infectivity after she added ether, this means it is sensitive to ether. And viruses that are sensitive to ether are enveloped viruses. So how do enveloped viruses exit the host cell? They can exit the host cell by exocytosis or budding. They do not really cause cell lysis, and this is why A, C, and D can be eliminated, and you're left with B, exocytosis or budding. So this button should be hyperlinked for you. You can try and click it. If it doesn't work, please use the link that I have here. Please take a moment to watch that video. So this is a video that will show you the life cycle of HIV, and you can see how the virus binds its receptor, how it enters the cell, how it replicates there, and how it exits. It's quite fascinating to watch. You don't need to worry at this point about the different details. and you don't need to worry about the last part of the video that discusses mechanisms of actions of drugs, but please go back to this video at the end of the course after you've seen how HIV causes infection and how it is treated, because I think it's a nice assimilation of all of these things that you will study in the course. And finally, as usual, this is my contact information. If you have any questions, please contact me, send me an email, and I'll try to answer your question. Thank you for listening.